Bismillahirrahmanirrahim. I am Sidra Mehmood and this is lecture number 35 of Nuclear Analytical Techniques. Today our topic is Instrumentation of Neutron Activation Analysis. The key concepts that are to be delivered in this lecture will be the introduction, the instrumentation of neutron activation analysis, and at the end we will discuss the calculation and the data processing involved in neutron activation analysis. So first of all, we will discuss the introduction of this classification of neutron activation analysis, that how this classification is being carried out. So first of all, the general procedure of neutron activation analysis can be divided into different stages like sampling, sample preparation, sample packing, irradiation and activity measurements, calculations of results, data processing and its interpretation. So first of all is the sampling. We will see that how this sampling is being carried out. Sampling is a technique in which the species of interest or the substance or the quality of any sample that we want to determine is first of all collected by using different methods, and then that sample or the substance which is to be analyzed is passed through different sampling procedures, and that is known as the sampling preparation, and then that sample is being packed by using techniques like sampling or sample packing. So how this sampling is being carried out is that that if for example we want to determine the concentration of different contents in water, and we have the water sample of a specific lake, then we will first of all categorize that a lake into different sites, and we label all these sites as A, B, C and D, and then we will collect the sample from all those sites by um, labeling those sites A, B and C. We will if add that sample into kivets, test tubes or any other bottles, we will also label those bottles or kivets or test tubes by that specific label that we have uh, done on that specific site. So in this way the labeling is being done and water is being collected, and again when when we put that a sample specific sample into that specific apparatus or test tube or cavite, we will also label it accordingly. Then we will take that sample to our laboratory and make different preparations of the sample uh, by using different sampling techniques, and we say that this is the sample preparation procedure. The sample is passed through different steps that like we have um, the sample which is being collected from the site. So that bulk amount of the sample which we have collected from a specific site is known as the lot sample, or we can say that this is lot, as it may be a gross or large amount of the sample, um, and if from that lot sample, if we take a some amount of sample, then we will consider that as a bulk sample, and when from that bulk sample if we consider a specific amount for the sampling preparation technique as to prepare sample in a lab, then we say that this sample is the laboratory sample. And out of that laboratory sample that we are using in lab, and we are only doing some analysis on that sample, then that particular or very small amount of sample is known as alicot. So this whole four-step procedure is known as sampling preparation technique, or simple we can say that sample preparation procedure. It involves the lot sample, the bulk sample, the laboratory sample, and the aliquot. Aliquot is the last stage at which is considered as the minute amount of sample which is used as the sample for the analysis. Then that aliquot is then packed by using different packing techniques, and then it is known this process is known as the sampling or sample packing technique. Then if our sample is exposed to some types of radiations, then it is considered as the irradiation of sample. And after irradiations, the activity or the amount or the quantity of the substance present in that amount of sample is to be analyzed by using different spectrophotometer or chromatographic or a different type of analysis technique, and those techniques are considered for the activity measurements. Further, at the end, we have obtained different results on the screen of the computer, and we obtain in chromatographic techniques different chromatograms, in spectrographic techniques different spectrums are obtained, and by interpreting the graphs of spectrum or of chromatogram, we can interpret different results, and those results will then proceed towards the conclusions and calculations, and the results are obtained by those interpretations of the graphical data. So at the end, the data processing and its interpretation is done for the further analysis. So of all these sampling and interpretation and conclusion and making of results, the irradiation and the activity measurements are the two main steps of neutron activation analysis, which will be discussed here. And those two main steps are mainly involved for the classification of neutron activation analysis. So these two steps, that are the irradiation and activity measurement of the sample, are mainly involved for the neutron activation analysis. So first of all, we will discuss the instrumentation of neutron activation analysis. The isotopic neutron sources can be used for some specific studies involving analysis of major constituents in bauxite or pyrolusite or alloys. So the neutron sources are used for the analysis of the major constituent present in different alloys, and mainly of the bauxite and pyrolusite alloys. And the neutron activation analysis mostly uses the sources having nuclear reactors. And the gamma ray spectrometry also forms the basis of neutron activation analysis. However, a method based on beta activity measurement has been developed for the determination of phosphorus via P32 radioactive tracer. Like we have discussed in previous lectures, that prompt gamma rays are majorly involved in the determination of neutron activation analysis, and they are produced when the neutron source hit or the neutrons target nuclei, then prompt gamma rays or radiations are being emitted. Meanwhile, beta rays are also generated, and they are also used for determining the activity of different substances. So this is the basic principle of neutron activation analysis, that how the neutron activation analysis are being carried out. The incident neutrons, when hit the target nucleus, the prompt gamma rays are being generated. Along with that, the beta particles are being emitted, and the delayed gamma rays are also generated along with the product nuclei. So these all the rays that are being generated are helpful in determining the neutron activation analysis of different substances. So the further steps that are involved in the instrumentation of neutron activation analysis is that the gamma ray spectrophotometer can be used, but it has the limited applicability when radiochemical separation is carried out. So the gamma spectrophotometer or spectrometer is used for the determination of the gamma rays, or to determine uh, how much the penetrating power of those gamma rays that are being radiated. But it has one drawback, that it has the limited applicability when radiochemical separation is carried out. So generally we use a detector which has much more sensitivity and much more selectivity than that of that prompt or gamma ray spectrophotometer. So generally an HPGE detector is used because of better performance in terms of energy resolution, peak to compton ratio efficiency, and linear response of pulse height and energy of gamma photons. So this HPGE detector is also known as high purity germanium detector. And these high purity germanium detectors are the best solution for precise gamma and X-ray spectroscopy. These high purity germanium detectors are very much very much more sensitive than that of the silicon detectors. They are much more efficient than silicon detectors as for radiation detection, due to the atomic number of germanium being much more higher than silicon, and due to low average energy necessary to create an electron hole pair, which is 3.6 electron volt for silicon and 2.9 electron volt for germanium. So due to energy, uh, low average energy required for creating the electron hole pair in case of germanium detector than that of the silicon detectors, germanium high purity germanium detectors are more conveniently being used than that of the silicon detectors. And the higher atomic number of germanium has a much larger linear attenuation coefficient, which leads to a shorter mean free path. So there are much more advantages of germanium detector than that of the silicon detector. That's why germanium high purity germanium detector is much more efficiently and conveniently being used than that of the silicon detectors. There are much other factors, like it has the better performance in terms of energy resolution, peak to compton ratio efficiency, and linear response of pulse height and energy of gamma photons. So due to these factors, it is much more applicable than that of the silicon detectors. And the Instrumentation of neutron activation analysis involves the HPE detector with all its electronic accessories as schematically shown in the figure in the next. Here is the complete instrumentation, that how this detector is being working. So first of all is the amplifier, and the sample is placed on the lead shield, where the gamma germanium detector is also present. It has high voltage power supply, and the amplifier is amplifying the signal. If the signals that are in very less amount, or we can say that if signals are very weak, then amplifier will amplify those signals and make sure that those signals will become much more intense, so they can be determined easily. So that amplified analog pulse is then moved on towards the analog to digital converter. Likewise, if those signals that are in very weak amount, or if they are very less sensitive signals and they are amplified, so the analog signal of that pulse is being digitalized and converted into digital converter. So the analog digital converter will convert the analog signals into the digital ones. So that again the digitalized pulse is then moved on towards the computer, and computer will then interpret those signals in the form of graph, and then printer will print that graph in the form of paper. So in this way we can have the signals of our required substance, or the amount or the quantity of the desired substance can be seen in the form of graph, or the signal can be interpreted on the piece of the paper. So how this whole interpretation occurs will be seen in that figure, that how this germanium detector is working to make high voltage to make those low intensity signals into digitalized high intensity signals in the form of graph, and is being printed on the piece of the paper, that how much intense those substance or those signals can be. The radionuclides are identified by prior calibration of the spectrometer using standard sources. In some cases, however, half-life of the radionuclide is also followed. So, in all these instrumentations, or in all these classifications or instrumentations, the radionuclides are being used. So they are used, and they are identified, and they are being detected. So they are working in different ways to examine the results and conclude the further conclusions. So they can be radionuclides can be identified by these calibration curves, and their half-life also affects the amount of the determination. If they have if they have very good half-life period, or we can say high half-life time, then they can be efficiently or conveniently being determined by using these techniques. So different parameters are being involved for determination of different substances. Like we have characteristic energy, most importantly the energy which is being used for the determination of substances, or for the detection of different elements, is the gamma ray energy. So in this, the gamma ray energy, E gamma, or which is being uh, denoted as KeV, uh, gamma energy, the half-life of a substance, T half, and other characteristics of common elements like isotopic abundance and cross-section, sigma, can be determined by neutron activation analysis. The typical gamma ray spectra of short and long-lived radionuclides formed in neem leaves along with its experimental conditions are shown in figures shown in next slides. Such spectra can be used for simultaneous determination of 25 to 30 elements in complex biological samples. Like if we have different samples in which different substance or different elements or components are present and we want to determine those elements or substances, then we make a different sample preparation techniques to make our sample to be analyzed by those spectroscopic or analytical procedures, and then different graphical spectra are being obtained, and those spectra are then interpreted to conclude different results. So this is the graph in which the chemistry or the composition of neem leaves is being analyzed, and the peaks of different graphs are interpreted to determine different components or the composition of the elements in that particular part of the leaf. So this is the graph which shows the composition of different elements present in neem leaves. So after different instrumentation and techniques, that how the instrumentation or sampling or interpretation is being carried out, the last step is the calculation and the data processing. As can be seen from the gamma ray spectra of irradiated samples of natural matrix such as neem leaves in figures shown in above slides, these are complex with closely laying multiple peaks. Like different elements are being present in different regions. We have different IR regions in which different elements are laying, different carbonyl groups lie in different regions, while aromatics and alkenes or alkynes lie in different regions. So these regions sometimes collapse with the other region, and it make difficulty in interpretation of different results having multiple peaks. So uh, those uh, those peaks or those multiple peaks that are laying one on the other, or those that are coinciding with each other, those peaks can also be interpreted by using the digitalized ways, or a computer can actually determine the percentage or composition of the substance by interpreting automatically all those graphs. The continuum under the peak is due to Compton effect from the gamma rays of higher energy and the background. So these peaks or the continuum, or we can say the irregularity in under the peaks, is due to the presence of the Compton effect from the gamma rays having higher energy. And the peaks show Gaussian distribution as the first contribution to the photo peak in the semiconductor detector is due to the statistical fluctuations in sharing the absorbed energy between ionization and heating the crystal network. It is not practical to analyze these spectra manually. Those spectra
can be analyzed, however, manually, but those spectra are digitally um, analyzed or interpreted by using the computer inner brain, in which the computer memory has saved thousands and millions of graphical representations of different substances, and it can automatically organize that data, and in this way the minute values of the elements can be determined by using that huge amount of data that is being placed inside the computer software, so that all the interpretations can be easily analyzed. And with the development of high resolution semiconductor detectors and 4, 8, and 16 Kelvin multi channel analyzers, gamma ray spectrometry, along with computers with suitable software, has become an important tool for the detection of natural and artificial radioactivity and activation analysis of complex samples. So, the complex samples, or the radio samples, or the activity of different radionuclides, or the radioactivity of different radio components, these all activities or measurements can be determined easily by using different spectrophotometric analysis. There are different multi-channel analyzers that are being discovered, or being used in different ways, so to enhance the uh, sensitivity of a particular instrumentation or particular detection. So different multi-channel analyzers are being used, along with different high resolution spectrophotometers, and different suitable softwares are being generated in many past few years, so to determine elements with high sensitivity and high selectivity. There are important steps in the computerized analysis of photo peaks, and those steps are the smoothing of experimental data, peak searching, selection of fitting intervals, peak energy calculation, peak area calculation. So these five steps are involved for the computerized analysis, for the calculation and data processing of neutron activation analysis. First of all, the data is smoothened, like if peaks are um, collapsing with each other, they are further smoothed, or they are zoomed, so that the collapsing, or where they are uh, adjoining with each other, those signals are being interpreted, so that the adjoining or the collapsing can be removed. So first of all, the smoothing of the experimental data is done. Then the peaks are searched, that which peaks are highly intense peak, as peak with sharper peak heights are considered as the intense peaks, and those peaks give the intense signals. So those peaks are being searched. Then the intervals are selected, and the peak energy calculations are done, and at the end the peak area is calculated. So in this way the data is being processed. The superimposition of photo peaks on each other, or the high Compton background, makes it difficult to precisely determine the net counts in the region of interest. And the region of interest is denoted as ROI. And the base area is mostly taken as trapezoidal summation of the average counts in each channel, giving the background over which the photo peak is situated, as illustrated in figure shown in next slide. So this is a particular peak in which the total peak area TPA is considered, and this is an intense peak in which the sharper edge is seen on the top, and all the area which is covering by the by this signal is considered as the peak area, and the height of the peak is considered as the peak height, and all these factors, the peak height and peak area, determine the amount and intensity of the signal. So this is all about this lecture. Now the assignment is determine how peak area is calculated for illustration of activity of any substance. Thank you.